The nitrogen cycle is a complex network of redox reactions mediated by a wide range of organisms, including plants, fungi, bacteria, archaea, and humans. In natural ecosystems, nitrogen is the nutrient most limiting to plant growth, as the vast majority of N is locked up in relatively inaccessible forms such as organic matter, and the atmosphere's huge reserve of N2 gas. Plants, however, acquire N from the soil largely in the most highly reduced form, ammonium, and highly oxidized form, nitrate, but are also able to influence microbial N transformations via the secretion of special compounds known as root exudates. One major source for plant-available N is biological nitrogen fixation, the reduction of dinitrogen into ammonia by diazotrophic N2-fixing prokaryotes. Here, root exudates can play a significant role, as exemplified in the case of flavonoids released from legume roots, which can trigger a complex signaling network between N2-fixing rhizobia and the plant, and result in the formation of root nodules that host the bacteria in a mutualistic endosymbiosis. Interestingly, it has recently been shown that root exudates from non-nodulating plants, such as maize, can influence root nodulation and hence N2 fixation in neighboring legumes. This discovery provides a critical mechanistic link to explain the synergisms and enhanced ecosystem productivity that have long been observed in legume cereal intercropping systems. Larger than all combined natural sources of fixed N are the anthropogenic additions, which derive from the industrial production of N fertilizers via the Haber-Bosch process, the large-scale cultivation of N2 fixing crops, and the combustion of fossil fuels. In other words, the activities of modern human have more than doubled the global N cycle. However, while anthropogenic N fixation is responsible for feeding half the world's people, it has also resulted in serious environmental and health problems. This is because the majority of N fertilizer applied to agricultural fields is not taken up by crops, but is lost to the air, water, and land via nitrate leaching and runoff, causing eutrophication, emission of nitrous oxide, N2O, a potent greenhouse gas and ozone-depleting compound, and the volatilization and downstream deposition of ammonia, which can contribute to indirect N2O emissions. However, root exudates can play a major role in the mitigation of these threats. In particular, biological nitrification inhibitors, BNIs, from root exudates of plants such as rice, wheat, and sorghum have been shown to target the enzymes mediating the nitrification reactions that oxidize ammonium to produce the soil-mobile anion nitrate, as well as N2O via so-called nitrifier denitrification. Many exciting studies on the influence of root exudates on the N cycle are now beginning to emerge and promise to contribute to the important issues of increasing agronomic N use efficiency and reducing harmful N losses to the environment, while boosting crop output to feed a growing human population.